So to select a workflow, you first need to select organism. Let's select measles virus. So this is the organism. We can debate whether virus is an organism, but nevertheless, it's here. If you click on it, you will see that there's one assembly, one genome sequence associated with this particular organism. To launch workflows on this assembly, we will need to click on Analyze button. Well, what that means is that, for example, when we will be doing variant calling, we have sequencing data and we are comparing this data against a genome to find variants. So in order to do this, you need to select genome first because you're doing this analysis in the context of a certain genome. This is what we're doing here. We selected this genome and we are going to be doing analysis in the context of that sequence. You can get more information about each genome and brc-analytics by using this View button. Let's go ahead and analyze. At this page, you have different choices of categories of analysis that you can run. So variant calling, RNA-seq, regulation, various other types of analysis, and we will be adding more. Let's do variant calling. If we expand this, we'll see two types of analysis. This is the paired and variant calling in a generic workflow, but here we have a more targeted workflow, which is specifically designed for viruses like measles. We're gonna go ahead and select that. So the next step is configure inputs for this workflow because it will need to know what data to take to perform the analysis. As I mentioned before, we already selected the reference. So workflow will be run against that reference that we selected here. Next, the workflow also needs to have access to gene annotations because once variants are identified, workflow will see which of the variants are inside genes, outside of the genes, and those are the inside genes, what effects they have on the coding sequence, on encoded amino acids, and so on. There's only one choice here, so let's click continue. So this is the step at which we actually select sequencing data, actual Illumina or element data set that we will use to find variants. You have two choices here. You can either upload your own data or you can pull data from the sequence-read archive. It's the same sequence-read archive that you visit if you go to NCBI or to ABI repositories. brc-analytics simply directly connects to these resources. brc-analytics also tries to pre-filter all data in SRA to only show you data sets available for this particular genome. Sometimes it makes sense, like right now. Sometimes it doesn't make sense, but you have full control here because you can always look at all data. It's just as a convenience feature, it pre-selected everything related to measles right now. Let's look at this. So the data, the actual data sets, each of these is an accession number corresponding to a set of sequencing reads. You can see that there are actually two files associated with each of them because the workflow that we're going to run is expecting paired end data. And on the left side, you have this panel which allows you to do various types of filtering. For example, it specifically only looks at paired end data because, as I said, the workflow expects this kind of data. But you can turn it off and on, and you can go into completely manual mode and select exactly what you want. So let's deselect WGS, which means whole genome sequencing, at this point, let's look at what studies are available for this particular organism. So if we click on study accessions, we will see a list of essentially experiments conducted by different labs. And for each experiment, we have a number of sequencing data sets inside that experiment. So these brc-ana IDs, they correspond to an experiment. If, for example, your lab is performing sequencing of a virus, and then it's going to publish these results, it's going to deposit them to NCBI SRA. And when you will be doing this, you will create the project for yourself, and your runs will be associated with this project. So it's kind of a container for storing data associated with a particular study. What I want to do here is I just want to select containers with the largest numbers of data sets. So 1, 2, 3. In the future, in the very near future, you will be able to sort this, so you don't have to scroll through this. But nevertheless, so we selected these three. You can see they're added as filters. And now this data set corresponds in the middle. You see these accessions corresponding to 225 Illumina runs. So let's select them all. Click Add. And this goes back to the workflow input controls. And I can just go ahead and click Launch in Galaxy, which will bring us to Galaxy interface. You need to, while BRC does not require user accounts, Galaxy does. So you can either log in with Google, or if you already have login, you can just go ahead and do that. Just log in like this. And at this point, you will get a launch interface. It's already pre-filled with all those choices that you made on the BRC side. So it knows what data, what sequencing data. It knows the gene annotations because we specified them. It knows what the reference genome is because we also specified. So you really don't need to do anything here. All you need to do is just click Run Workflow. And this will bring you to the workflow invocation page, and you will be able to trace the progress of your workflow as it goes through all the steps. And there is no such thing as real-time genomics, so it's going to take a while. This particular analysis can probably take an hour or two.